Hello everyone! Today we continue to study general embryology. We have learned germ cells and fertilization. Now we will learn formation of blastocyst and implantation. We have two sections. Section 1, cleavage and formation of blastocyst. Section 2, implantation. Once the fertilized ovum is formed, it begins to move towards the uterus and undergo cell division. Because the daughter cells are surrounded by the zona pellucida and there is no growth process in the interface, only the cytoplasm of the original fertilized ovum is continuously divided into the daughter cells. So, with the increase of the number of cells, the cell volume gradually becomes smaller. This special mitotic process of a fertilized ovum is called cleavage. And the daughter cells produced by cleavage are called blastomere. On the third day, there were 12 to 16 blastomeres, which formed a solid embryo with the appearance of a morula. Cleavage is a process of continuous acceleration in 12 hours after fertilization. Single cell, 30 hours, 2 cells, 45 hours, 4 cells, 72 hours, 16 cells. This is a picture of morula. The shape of the embryo on the third day after fertilization is similar to that of morula. In this slide, we look at the changes in cleavage and blastocyst formation. It can be seen that the red ring structure is the zona pellucida, which existed all the time before the formation of the blastocyst. With the formation of the blastocyst, the zona pellucida began to degenerate and disappeared after the formation of the blastocyst. On the fourth day, the morula entered the uterine cavity, and its cells continued to divide. When the number of blastomeres reached about 100, there were several small cavities between the cells, which gradually merged into one cavity filled with liquid from the uterine cavity. At this time, the zona pellucida dissolves and the embryo is vascular, so it's called blastocyst. The center of the blastocyst is the blastocyst cavity. The wall of the blastocyst is composed of a layer of cells, which is related to the absorption of nutrients. It is called trophoblast. A group of cells located in one side of the blastocyst cavity, called inner cell mass, have multiple differentiation potential. The trophoblast at one end of the inner cell mass is called polar trophoblast, also called embryo trophoblast. It covers the surface of the inner cell mass, and the cell body is slightly larger than the trophoblast cells in other parts. The polar trophoblast is related to the implantation of blastocyst. These are early and late blastocysts. It can be seen that there is a residual zona pellucida outside the early blastocyst, but not in the late one. After the complete formation of the blastocyst, there are 3 parts of the structure. The outer part is a flat layer of cell, called trophoblast. The inner part is a cavity, called the blastocyst cavity. The cubic cell near one end is called inner cell mass. After proliferation and differentiation, these cells finally develop into human body. This is a picture of early and late blastocysts. We can see the difference between them. the zona pellucida disappeared. The trophoblast cells became flat and the blastocyst cavity became large. This picture shows the locations of different stages: ovarian ovulation, fertilization in the tubal ampulla. As the fertilized ovum moves to the uterus, it begins to cleave. 2 cells, 4 cells arrive at the corneal uterine on the 3rd day, and the morula is formed. On the 4th day, the blastocyst were formed and embryo entered the uterine cavity. I think you can see the structure of each part of this picture. This layer of columnar cells is a single layer of columnar epithelium of endometrium. The vesicular structure is the blastocyst. The outer layer is the trophoblast, and the inner group of cells is the inner cell mass. It can clearly see that polar trophoblast cells adhere to endometrial cells.